Hi, I'm Lauren Coombe from Canada's Michael Smith Genome Sciences Centre at BC Cancer in Vancouver, Canada. And here I'm going to describe my poster, LongStitch, High Quality Genome Assembly Correction and Scaffolding Using Long Reads. So LongStitch is a de novo assembly, correction, and scaffolding utility which uses long read evidence and runs in up to 3 steps. So given a set of long reads and an assembly that you want to improve upon, there's a round of assembly correction with Pigment long, and up to 2 rounds of assembly scaffolding with NT-Link and ArcsLong. NT-Link being a newly developed long read scaffolder that we're introducing within LongStitch. So before diving into the algorithmic details of LongStitch, first starting with some background about minimizer sketches, which is something that's pretty important within the NT-Link algorithm. So minimizer sketches reduce the computational cost of sequence data storage and manipulation, and they're pretty widely used in various bioinformatics tools such as Minimap2, also our own NT-Join. So the idea is that instead of computing all of the k-mers on all of the input sequences, which can be quite costly, use a particular subset of k-mers. So in generating our minimizer sketches, for each window of W adjacent k-mers, we compute the hash values of each k-mer using NT-hash, and then we assign the Windows minimizer to be the smallest hash value. And repeating that over the length of the sequence generates an ordered list of minimizers per sequence that we can use to map the long reads to our assembly. So now diving into some details of the NT-Link algorithm. So the inputs are your long read evidence and the assembly that you're hoping to improve using those long reads. The first step is generating these ordered minimizer sketches that I just described. So here I'm representing minimizers with those circles. And then those sketches are used to perform a lightweight mapping between the long reads and the assembly. And this is done for each long read. So starting with that bottom long read in the third step, you can see that the identical minimizers are indicated by those dotted lines. So from those minimizer mappings, this long read is giving evidence that A should be followed by B in the positive orientation, B followed by C, and also transitive information that C should come sometime after A. And so this information is tallied for all of the long reads. And then given all of that information, it's represented as a directed scaffold graph where the nodes are oriented contigs and there's edges created between the contigs in the pair with the edge weight being the number of supporting reads. That directed scaffold graph is then input to a scaffolding layout tool called Abyss Scaffold, which is part of our Abyss suite of tools to generate your final ordered and oriented scaffolds. And just another detail that NT-Link does perform gap size estimation, and that's done using the positions of minimizers in the long reads compared to the draft contigs. And of course, this gap size can be negative, and that would happen if the contigs overlap. So as well as introducing our new NT-Link long read scaffolding tool, within LongStitch we're also introducing Pigment long and ArcsLong, which are adaptations of our previously published tools Pigment and Arcs, which were originally developed to use linked read evidence, and we've now adapted them to use long reads as well. So the main change for these tools was the addition of a data preprocessing step where we generate pseudo-linked reads from the input long reads. So linked reads need an associated barcode for each read pair, and in this case, each read pair that comes from the same long read is given the same barcode. And then those pseudo-linked reads are input to Pigment long and ArcsLong. There's also some other small changes that were made within those tools, such as different parameterization, using Minimap2 in Pigment long instead of BWA, but this data preprocessing is really the main change. And here we're showing the result of running LongStitch on some human data. So we first obtained Oxford nanopore reads for 3 different human individuals, as well as some short read sequencing data. So we generated a short read Abyss assembly and a long read Shasta assembly for each of these individuals, and improved upon all of these assemblies using LongStitch. So first looking at the contiguity and correctness plot, on the y-axis we have the NGA50 length, which is a measure of contiguity and correctness. The x-axis is the number of quast misassemblies. The white dot is the stats for the baseline assembly. Blue is the stats for LR-SCAF, a comparator long-read scaffolder. And then you can see the stats for the different steps of LongStitch and increasingly dark shades of orange. So from the white baseline dot, you can see a reduction in misassemblies following Pigment long, and then an improvement in the NGA 50 length following the NT-length scaffolding step. And these two steps are the default mode of LongStitch. If you want to do additional scaffolding, you can get further gains using ArcsLong. So you can see here for 5 of the 6 runs, running LongStitch achieved a higher NGA 50 length as compared to LR-SCAF. And in all cases, LongStitch led to fewer quast misassemblies than LR-SCAF, which we think is really important for downstream analyses. Looking at the benchmarking, looking at the peak memory versus time, running the default LongStitch pipeline was faster than LR-SCAF in 5 of the 6 runs, and both of the tools used a pretty similar amount of memory. We then visualized the assemblies from the previous slide with Jupyter plots. So in these plots, large misassemblies are very evident as interrupting ribbons. So this is showing the results for NA24385. You can see in both the short and the long read comparisons that LongStitch plots have fewer interrupting ribbons, also indicating that there's fewer large-scale misassemblies. And finally, to conclude, LongStitch is a scalable assembly correction and scaffolding utility. It leverages the rich information in long reads to generate high-quality final genome assemblies. If you want more details about LongStitch, we have a paper available in the bioRxiv, and our tools are available on GitHub as well as through Bioconda. Thank you very much for listening.